The next organelle we're going to talk about is a chloroplast. And chloroplasts are going to be found in plant cells and also in algae. The job of chloroplasts is to capture light energy and take that light energy and store it in chemical bonds of glucose. So let's go ahead and write chloroplast. Again, the job of a chloroplast is take the energy of sunlight and store that energy in the chemical bonds of glucose. Now, chloroplasts are typically round, however they can be oval. This is going to be the outer chloroplast membrane. The chloroplast also has an inner chloroplast membrane. And the space between the two membranes is the intermembrane space. Within the chloroplast are stacks of membranes, stacks of sacs of membranes. Lots and lots of these stacks. I'll draw just a couple of these stacks. The membrane here is referred to as a thylakoid membrane. This thylakoid membrane is actually going to contain chlorophyll molecules. These are small proteins that are embedded in the thylakoid membrane. And again, the more chlorophyll proteins I have, the more light energy I can capture and store in the form of glucose. So, what we want here, just like in the mitochondria, is lots of membrane, lots of surface area. And the chloroplast achieves that in a slightly different way. Instead of having the inner chloroplast membrane very convoluted, we're going to create lots of surface area by having stacks and stacks and stacks and stacks of these thylakoid membranes, like a stack of coins. And by having so many stacks, we create a large amount of surface area. Now, within the space here, within the thylakoid membrane, is a space called the thylakoid space. And it's a small space, and we're going to use that to our advantage. We're going to pump hydrogen ions in this small space, in the thylakoid space. Lots and lots and lots of hydrogen ions. This high concentration of hydrogen ions is going to represent, again, potential energy. We're going to store it, and later we're going to release that energy and store it in the chemical bonds of glucose. Each one of these stacks of thylakoid membranes is referred to as a granum. And each chloroplast has many, many, many grana. Again, there's a jelly-like material in here. We're going to call it the chloroplast stroma. And what makes it different from the nucleoplasm, different than the mitochondrial matrix, is it contains different proteins dissolved in it, giving it a different function. And that is going to be it for the chloroplast. Lastly, I want to tie together a couple organelles. We're going to start with the nucleus because that manages the entire cell. So I'm going to go ahead and I'm going to write nucleus. Now the nucleus contains the DNA, which has the information needed to make every molecule inside the cell. Now, the proteins that the information actually encodes are created in the cytoplasm, outside the nucleus. So we've got a bit of a problem. The information needed to make the proteins is inside the nucleus, and the machinery to make the proteins is actually in the cytoplasm, outside the nucleus. So nature came up with a way around this. Let's copy the information in the form of a messenger RNA molecule. So we're going to copy that information into messenger RNA. So we're going to copy the information of DNA into messenger RNA. And then that messenger RNA is going to leave through the nuclear pore and go into the cytoplasm and bind to ribosomes. These are the factories that make proteins. And they come in two varieties: free ribosomes floating around in the cytoplasm, or bound ribosomes that are actually attached to the endoplasmic reticulum. Now, because there are so many small ribosomes attached to the surface of the endoplasmic reticulum, which is a series of membranes just folded up, if you look at this under a very powerful microscope, it actually looks rough. And because of that, the scientists decided to call it the rough ER, or the RER for short. The ribosomes actually take the information in the messenger RNA and take amino acids that are floating around in the cytoplasm and put them together, connect them up in the correct sequence. And then as they connect them up, the protein actually begins to fold and take its three-dimensional shape. Once the protein is made, it's going to go to another organelle called the Golgi Apparatus, or Golgi complex. Capital G because it's named after Dr. Golgi, who actually discovered this structure. The Golgi is very important. It does two major jobs. One, it modifies proteins by removing amino acids and by adding short sugars or carbohydrates to the amino acids. This changes the function of the protein slightly, puts the finishing touches on the protein. Also, the Golgi apparatus acts as a post office. Proteins come into the post office, and then the post office determines where those proteins are actually going to end up. So it's responsible for determining the destination. There's essentially four destinations where the proteins can actually end up. They can end up secreted from the cell, so they'd actually be released through exocytosis. They could be embedded into the cell membrane, become part of the cell membrane. They could become part of the cytoplasm. Or finally, they could become part of an organelle. And those are the locations that the proteins can actually end up.